Hi, my name is Heidi Hanson. I'm an assistant professor at the University of Utah. Today I'll be talking to you about new methods for identifying complex disease in families and linking them to their etiological roots. The main point of my talk is going to be to discuss how combining data from a vast number of sources with machine learning methods can help us make sense of the interdependent genetic and environmental factors that shape health. I'm going to start off by walking you through a few of the ways that I'm using to triangulate genetic and environmental underpinnings of cancer, with the hope that they will be able to be translated into the clinic for improvements in prevention, treatment, and recovery. The data for my study will be coming from the Utah Population Database. The Utah Population Database is a unique data set that combines data from multiple sources to facilitate life course research. However, it's not unreasonable to expect that a data set multiple data sets like this will exist in the near future, maybe 5 to 10 years. And so these methods will be applicable to many different studies and hopefully help us identify etiological factors that lead to disease. At the end of this talk, you should know how machine learning methods can be used to understand familial patterns of cancer, and how you can start to build your own resource for measuring environmental exposures across the life course. The Utah Population Database, as I mentioned, is a unique resource. It contains data on over 35 million demographic and medical records and contains linkage IDs that allow us to link to an additional 60 million records at different institutions. One unique component of the database is the deep family history linkages, which go up to 18 generations. Utah Cancer Registry records from 1966 to the present and cause of death certificate records beginning in 1904 are the key elements that lead to our phenotype, so that those are measures of cancer are coming from those records linked to family history. So I want to pause here for just a minute to let everybody know that we take data ethics and oversight very seriously at Utah Population Database. And I take it very seriously within my own research as well. So at the institutional level, we have two separate bodies that review the studies that are coming through and using the Utah Population Database. We have one that's called the Resource for Genetic and Epidemiological Research, and that is consists of one member from each of our data contributors, and they all review the studies prior to the data being released. All studies are also required to undergo institutional review board, and only the minimum amount of data necessary is ever released to the investigator, and all data may stay on our machines or our servers so that we can control the data at all times. At the individual level, my lab conducts regular ethics meetings, data ethics meetings, where we discuss many different topics, including the importance of considering the bias that are in our research, selection biases and other data elements, and then how we can make sure that the research that we're putting out to the community has been well thought out and is beneficial. So for my first story, I will be walking you through a classic measure of family history of disease and describing how machine learning can inform pedigree selection for future research, investigating the genetic and environmental determinants of cancer. Family history of disease has historically been important for the identification of genetic causes of cancer. Searching for families with higher than the expected risk of a single cancer can reduce the noise in a complex phenotype or definition of disease, such as cancer, and increase our power to find associations. Using information about cancer patterns in the family can narrow our search and lead to important discoveries. However, many of the highly penetrant genes have been discovered, and this method is no longer yielding powerful results. Genome-wide association studies have also come up short. So I would like to discuss new ways that we can think about utilizing the information that comes from family history of cancer. So this is a picture, this is a focused in image of Surratt's Law Parade, and I edited the picture so that you could only see the red parts. And what you see is by only looking at a single color at a time, we're muting what's actually going on, and we're not looking at the full painting and seeing everything and all the information that exists within that painting. However, if we incorporate all the information or more information, we can start to see patterns in in this painting, and in our case in the data, that may be informative for us to understand what exactly is going on within these families, thus reducing the noise in the data and improving our ability to discover genetic and environmental causes of disease. So this is an example of how we can use the information that we have in our database. BRCA1, BRCA2 are two known genetic mutations, and we currently know that those mutations predispose individuals to more than just breast cancer. And each of those mutations carry their own unique fingerprint within the data that allow us to see what kinds of cancers that they might be predisposed to. So when we're looking at family history, expanding our view to look at multiple cancers may increase our information about our knowledge about the underlying etiological factors of disease. So the classic approach to investigating the risk of cancer in relatives of bladder cancer patients, which is one of the cancers that I'm interested in researching, is to run separate regressions for each cancer of interest. This increases our knowledge about the bivariate relationship. So if I have urothelial cancer, what is the risk for urothelial cancer in my first, second, and third degree relatives? And that tells me a little bit about the genetic and environment underpinnings of disease, just as Dr. Patel explained in his talk. And we can expand that and say, okay, if I have urothelial cancer, what's the risk of kidney cancer in my relatives? And we gain information about the relationship between diseases. However, this approach really identifies relationship between two diseases and doesn't look at the overall patterning of diseases within families. And that information may give us more knowledge about their underlying etiological roots. So how do we see the bigger picture? Well, machine learning is an approach that allows us to discover previously unknown patterns in our data and group families with similar patterns together. Identifying families with similar patterns of cancer clustering can inform studies of genetic and familial determinants of cancer by reducing the noise in our definition of phenotype and helping us identify what factors are contributing to disease. So, as Dr. Patel discussed in the question and answer section, selecting the right method is something that can really help us and is a really important question when we're asking, when we're starting to use these complex methods. So how do we actually go about doing that? Well, first, we need to figure out what we're trying to understand. It may seem like a simple given thing, but a lot of times it takes a little bit more thought than maybe we're thinking of. So there are two main approaches for machine learning, and the choice can be reduced to whether you want to develop a predictive model for a known outcome, such as mortality, which is what you're seeing on the left-hand side as a supervised learning model, or if you want to identify patterns in the data that allow you to generate hypotheses. So taking you back to the Surratt picture, can we find patterns that say, okay, this is something like a face, or this is something like a shoulder? That gives us information about what we see in the data, and hopefully, in our case with cancer, allows us to see if the patterns that we are seeing are due to environmental components, genetic components, or more frequently, the combination of both. Another question we really want to ask ourselves as we move forward in this type of research, and this is probably the most important question that maybe people don't spend quite as much of time thinking about, but what do my data really look like? You need to understand the structure of your data in order to pick the correct method, and it is very important. So how much missingness is in your data? Are there biases in your observations? Do you need to normalize data to make the method work appropriately? Do you have mixed data types? Are you expecting to see linear versus nonlinear relationships? And how is sampling affecting what you're actually seeing within your data? In addition to asking what my data look like, you need to ask yourself, what are the nuances of the method that you're choosing? There are many different packages out there that will give you a result at the push of a button. But you should take the time to understand the parameters of your model and figure out how to determine which model best fits your data. And this is something that you should be very thoughtful about. In my case, one of the things that we had to think through is how do we identify the number of clusters, or the K, that are appropriate for our model. And there are many different methods out there. we took an approach of combining information from those multiples out there, which allowed us in the end to find 5 separate familial clusters of cancer, bladder cancer clustering. And this allowed us, these clusters will provide a foundation for our future studies that we are planning to link etiological factors to cancer risk and survival. And you can see that there are many different patterns in the data, and that there's more than just a single bladder cancer to kidney relationship, but some of these relationships are much more complex, and being able to have a broader lens and incorporate that complexity into our research will hopefully help us understand cancer better. So I have taken you through the process of increasing our ability to identify patterns in familial data using machine learning methods.